You're halfway.
Hello, everybody. Obviously, I got dropped. Can you all hear me?
Yes.
Okay. Yep. All right, we got one more slide of horizontal gene transfer, and then we're going to talk a little bit about HIV. As I said, obviously, I lost my internet connection. Is being recorded. It takes a while for me to reboot my computer and get everything back up. Sorry for the delay. We were talking about Denisovians, another species of another individual in the genus Homo. We talked about Tibetans, how they have an allele that's identical to that Denisovians have, which allows the Tibetans to live at very low partial pressure of oxygen, only 40% of what is found at sea level. It does so by having low amounts of hemoglobin. Most of us, when we're exposed to, those of us that don't have the allele that the Tibetans have, when we're exposed to low partial pressure of oxygen, we tend to respond by the production of more hemoglobin. The problem with that is the increased production of hemoglobin increases the viscosity of blood, which causes mountain sickness, high altitude mountain sickness. There's a reason why people wanting to climb large mountains have base camps to acclimate over time, so that it's a stepwise process and the body can handle increased production of hemoglobin. Increases blood viscosity, which causes all kinds of problems, heart attacks, strokes, things like that. One of the traits that's been observed in Tibetans, presumably due to this allele, is that they have a more elaborate blood vessels. That is, they have more. It's a more elaborate network than what those of us that don't have that allele. And another observation, but it's not clear it's linked to this particular gene, is that they tend to have a very high birth weight, and nobody really knows why. These individuals, Tibetans, clearly are not susceptible to altitude sickness. They think that they adapted in roughly 3,000 years, which would be in conflict with the idea that the allele came from the Denisovians. It's also true that we know that some populations, like I said, in Australian Aborigines, Filipinos, and whatnot, some new HLAs were obtained from mating with the Denisovians. These are antigens that are involved in the immune response. Okay, so that's the end of the material on horizontal gene transfer. I think you can see that it has played a significant role in the process of evolution, not just of unicellular organisms, but of our own species. Now we're going to talk about HIV. HIV, as I'm sure you're aware, can lead to the disease known as AIDS, acquired immune deficiency syndrome. There's a receptor, CD4, that is required for infection of the most common version of HIV. There's actually two HIVs, one derived from a monkey, the other derived from a chimp. The one that is spread throughout the world is the one derived from chimps, HIV-1, and the most common strain is HIV-1M. That strain in particular requires attachment to the receptor on our cells of CD4. Come on. It's a glycoprotein expressed on the surface of helper T cells, macrophages, and others, all those that are susceptible to infection by HIV. Yep, it's the receptor to which HIV-1 binds. The virus itself has an envelope protein referred to as GP120, which binds to this receptor. That's the initial step in infection by HIV. The envelope protein of the virus undergoes a conformational change and actually recruits other receptors. So it's more than just one receptor, but it starts with CD4. Eventually, after enough receptors have been recruited, there's actually a fusion of the viral membrane with the host membrane, which allows for the virus to dump its cellular contents, the capsid, inside our cells. This is an illustration of the life cycle. You can see CD4 receptors are indicated. There are other receptors which are not shown on this illustration. The capsid, which is a protein coat that encapsulates the genetic material of the virus, gets injected into or gets through fusion. It's not really injected, it's through fusion. Ends up inside the cell, the cytoplasm. The RNA HIV virus has 2 copies of its genome present, as well as 2 copies of the enzyme reverse transcriptase. One of the first things it does is to take its RNA viral genome and make a cDNA copy of it. And once both strands are copied, it can now integrate itself into the host genome. And that's when all the problems begin. It can replicate itself through transcription, producing literally thousands of copies, as well as the proteins that are required, the different proteins for the capsid and the envelope and whatnot, GP120, till you get back a functional virus. And just as in entry, you have fusion, and in this case, release of the virus to go and infect other cells.
Are the promoters for the involving in like replicating these included into the RNA that is converted into cDNA?
Yes, it carries everything it needs, basically. It's not like enhancer traps where its expression level is going to be dependent upon wherever it inserts itself. It carries all the DNA, ultimately DNA information that it needs. Starts off in the RNA and then goes to cDNA. The level of CD4 is used to monitor the progression of the HIV infection. I'll show you in just a second. Can cause direct killing of cells. The virus can, that is. CD4 levels are going to go down over time, and that's what's monitored. Can induce apoptosis, where cells know something's wrong and they commit suicide. Could be destruction by cytotoxic lymphocytes in our blood cells, destroying it. But the consequence is CD4 levels drop as the infection continues. Yeah, used to monitor the progression of the disease. Here you can see an illustration of this is CD4 levels prior to infection with HIV. You have initial infection with HIV in the red. You see it spikes very early after infection. Then the host cell's ability to control it starts to drive down HIV. At the same time, CD4 is dropping in amounts. You get some recovery of CD4. HIV slowly starts to increase. CD4 continues to decline. Eventually, you become susceptible to opportunistic diseases, particularly pneumonia and things like that. As HIV concentrations go up, CD4 goes down, and ultimately death. You can see the timeline on this particular illustration is about 10 to 11 years after infection. This is an illustration of what the virus looks like. You have the GP120, so-called docking glycoprotein. That's the one that's going to interact with CD4, the receptor on our cells. You have some other membrane proteins. You have a lipid membrane, which allows it to fuse, the virus to fuse with our lipid membranes. You have a capsid, which is a protein coat, basically, that encapsulates the viral genome. You can see there are two copies, two RNA copies, and two reverse transcriptase, one attached to each of the two viral genomes. Acquired immune deficiency syndrome, AIDS, was originally called GRID. Does anybody know the origins of the term GRID? Okay. Well, I wish we were in a classroom. It actually stood for gay-related immune deficiency. If you remember the history of HIV, it was discovered by a physician who noted that a lot of young males, gay males, were coming down with a rare form of skin cancer that was normally only seen in much older males in the Mediterranean area. But here you had California